In this lecture, I will discuss some experiments by which scientists have discovered antibody and the structure of antibody. So the first experiment by which one scientist have discovered antibody performed by Kabat. He took a rabbit and injected the rabbit with a antigen called ovoalbumin injected. After a few days, he took serum from the same rabbit and divided the serum into two parts. In one part, in the serum, he added ovoalbumin, and another part of the serum, he do not add anything. After this two serum, he performed a gel electrophoresis, and he found from this serum, he found 4 peaks. One is very high, another is small, another is very small, another is slightly high. He called this is albumin because albumin is present in our body in the most high concentration. So this is albumin. The next peak he called alpha because in Latin alpha is the first letter. Then he called it is beta, the second letter, and it is called gamma. When he performed gel electrophoresis with this portion of serum, he found a very interesting fact. He found that the albumin level was same, but the alpha antibody slightly down, the beta portion of the protein slightly down, and the gamma region is very down. He found smaller falls, not very high fall, but smaller fall in alpha and smaller fall in beta, but a huge fall in gamma. Now the protein level is here. So he found a very high fall in the gamma, and he concluded that this portion of this protein is actually antibody. Why? Antibody binds with antigen, it can precipitate down. So this albumin do not falls because it is not antibody, but slight falls of alpha and beta, it denotes that antibody can be in this group also, but a huge fall in the gamma protein means this gamma portion actually denoting the antibody, as because when serum that contains antibody and the same antigen which is actually injected in the first place, ovoalbumin, the same antigen is adding this antibody actually complex with this antigen and precipitate down. This is called immunoprecipitation. So this is immunoprecipitation. So the reason behind this fall is the immunoprecipitation. So the antibody is called gamma globulin. As you know that there is some falls in alpha and beta, so there are few antibodies that are also fall in the alpha and beta globulin, but the majority of antibody falls in the gamma globulin. Like all IgG antibody falls in this gamma globulin. So you can see how scientist Kabat has discovered the presence of antibody in the serum. The next 3 experiments by which we have known the structure of antibody, one is using papain enzyme, the third is using pepsin enzyme, and the fourth is using beta-mercaptoethanol. This is actually reducing agent. So let me draw a antibody structure. A antibody looks like Y. So by performing these 3 experiments, we have known this. The antibody looks like this. So antibody, this region is called heavy and this region is called light. This is heavy chain and this is light chain. When we digest the antibody with papain enzyme is present in the papaya fruit. When this enzyme is actually reacted with antibody, this enzyme cut the antibody in these 2 regions and produce 2 similar weight, molecular weight molecule and 1 different molecule. Scientists have found that the one different molecule can be easily crystallizable. So they called it Fc region. So if we digest the antibody with papain, we will find 3 fragments. So the result of this digestion of papain is a 3 fragment, 2 with similar weight and another is that is called Fc region, because this region can be easily or spontaneously crystallizable. So this is called Fc region, fragment of crystallizable, and the 2 region is called FAB region. This FAB region or antigen binding region was known by performing this experiment, digesting with the pepsin, because when we digest this antibody with papain, the 2 fragment that is produced called FAB cannot bind with antigen. You have to remember this. This FAB, both this FAB cannot bind with antigen when it is FAB conditions. So by performing this papain experiment, scientists don't have know that this region is actually responsible for antigen binding. But by performing the digestions with pepsin, they have found that this region is actually responsible for antigen binding. How? I am showing this. When we digest the antibody with pepsin, the enzyme cut the antibody in this region, not this region, in this region. So now the antibody becomes 2 fragments and the remaining portion of the Fc now being digested by the pepsin. So pepsin will produce only a single fragment, 1 fragment. This region is called FAB bar 2. This region can bind with antigen. So from this experiment using pepsin, scientists have known that the 2 segments that is produced during the papain are actually responsible for the antigen binding of the antibody. So this region is called FAB, fragment of antigen binding, and this region called Fc region. This Fc region is responsible for the effector function of antibody. But if we use beta-mercaptoethanol, disulfide bond will be break and 4 fragment or 4 separate chain will be produced. So now you can see this is one light chain, this is another light chain, and this is heavy chain, another heavy chain. So by using beta-mercaptoethanol, scientists have found 4 fragments. They have found 4 fragments and they have understood that 2 similar lightweight chain and 2 similar heavyweight chain